All right, good morning. My name is Deepak, and it's a pleasure to be talking to you today about how do we really think about, if we're in inning one, I guess we're like in the warm-up still then in synthetic biology and organoids. But sorry, Eric, CAR T-cell was the first out of the gate, but we're coming for you. So one of the whole things is, how do we think about looking at human disease? When we actually consider just these last 2 years with SARS-CoV-2, so much of learning about COVID was actually observational. We cannot test on patients as this disease is progressing, so we need to think about how do we recapitulate model systems. And the first out of the gate, as always, you're going to actually think about your animal models. You're going to think about what can we learn from various animal models that recapitulate human physiology. As everyone knows in this room, a mouse is not a human, but a mouse is a mouse, and there are some things we can learn. And we can go up and down the various animals, and maybe we can actually recapitulate heart physiology in a zebrafish. Maybe it actually turns out that the stomach of a pig looks really similar to a human stomach. But from there, we've actually kind of outlined the other dimension, which is maybe we don't need the entire animal. Maybe we can actually learn a lot of just the underlying biology with a single protein, with a single model system. And that leads to the development of these immortalized cell lines. Some of them are going to be from cancerous cells, some of them are going to be coaxed in a lab using very specialized media that really is not the wonderful burritos that we just ate for breakfast. Like DMEM, tasty, maybe. So there's a need to really think about how do we study disease and what can we as a synthetic biologist, what can we do as communities leverage the power we now have with molecular biology. And so for today, I'm going to introduce the concept of an organoid and what an organoid can actually enable. So for the purpose of this talk, organoids are iPSC-derived multicellular bits of tissue that have some function related to the organ they're derived of. And the hope is at some point we drop the oid and we actually are just creating organs. So in synthetic biology, we think about what we can do by thinking about the state of a cell. Every cell is going to have a collection of transcripts, proteins, metabolites, and can we interrogate that state using different modalities? And so to really set the stage, we have to think a little bit about how we got to kind of the ability to make circuits. We started with primary cells. These primary cells were grown, specialized media, isolated from patients. Often they die. We've been starting to mix cells together, so now we get 2 different cell types or 3 different cell types from various patients. I start engineering encapsulation. I start engineering kind of how we grow them, and we're starting to make like little balls of cells. From there, we've started to get stem cells with Yamanaka factors where you can make spheroids, and now you have a little bit of differentiation. So spontaneously, if I put some stem cells down, I will get all sorts of cells from it. And now we really mixed in, particularly in the Boston area, the use of micropatterning, the use of scaffolding, the use of 3D printing technologies to think about the fact that a 2-dimensional cell culture is not actually going to recapitulate structure and functional aspects of an organ without introducing hydrogels or other elements. But we still have a wide range of problems to address if the hope is to really get to an iPSC-derived, multicellular, patient-specific organoid that is capable of either regenerative medicine with implantation or even just lower level hanging fruits such as drug toxicology, drug development, and metabolism. And these challenges are really enlightening is we need to look at gross anatomy to understand what cell types are even in your tissue before we can start thinking about how do we design that. And so we need to figure out the spatial and 3D organizations that we need to recapitulate. We need to think a little bit more about how many patients do we have to sample? What are the actual differences in organ structure from person to person? And what are those variations in actually the metabolism of each patient? And there's plenty of challenges that we're starting to address. How do we actually mature an organoid, I think, is the biggest question, and that's going to be the rest of my talk today. So as a synthetic biologist, if we can interrogate a cell, and we can actually tell what the proteins, what the RNAs, what is that state of the cell, we should be able to design a genetic circuit that looks at that state and tell me where I am and where I'm going. From there, I put that into a patient-derived cell, and I actually just expand that iPSC population. So we might use automation, we might use full GMP compliant facilities that thankfully CAR T cells are making for us. And from there, we have to undergo a symmetry breaking operation. So now, these cells have all of the different, maybe germ layers, maybe the different cell types, and in turn, with some developmental development, we will get to this intact organoid. One of the hardest things, though, about stem cell biology is we are still learning all of the features that a stem cell can do. So classically, if we think about our stem cells, we might look at it like this. We have a stem cell that goes through some series of steps and eventually becomes a terminally differentiated cell. And that is a canonical Waddington model that has been around to really anchor ourselves in differentiation biology. However, stem cells are really tricky, and we're learning this every single year, where there are multiple paths that are just really not sequential. We have graded bifurcation events where cells can actually, depending on the signals given, actually really bifurcate into two distinct populations. We can have direct differentiation of cells to a terminal state that is already ready to go for testing or that cell type. There's all of the pluripotent programming that we're going to end up using when we go at Yamanaka factors and patient derivations. We can have different niche requirements where cells won't differentiate correctly unless you have the partners that you need by it. And there's always direct conversion. So all of these portray then, if we're going to interrogate by writing a genetic network in a circuit to differentiate stem cells, we need to really think about all of the different paths it can take. One of the hardest things is what happens if differentiation gets stuck? And this happens more often than you think, is in the lab, kind of state of the art is you have this stem cell population, you're adding growth factors to the media, you have no spatial orientation because you just have a series of cells you're trying to coax through these steps, but what happens if a step goes wrong? And so from an engineering consideration, we need to start thinking of the following parameters. When do I intervene with a stem cell? And what timings do I use? Is this transient? Is it permanent? How do I do it? Does this need to be at the RNA level? Can it be at the DNA level? Is it actually a protein phosphorylation cascade that we need to be manipulating? And how specific do we need to be? Is it just a single cell getting stuck? Is it a population of germ layers that just aren't being developed? And is it some spatial orientation with morphogen gradients where we're recapitulating development? Or is there actually a little easier engineering shortcuts, if you will? And so the way we've been approaching this problem here at MIT with the SynBio Center is we think about these as a series of steps. And these steps are not necessarily a binary decision tree, but rather a what is the cell's current state, and can we take that state and just kind of progress it along. And so for instance, if I wanted to just make another solid organ, in this case a beta cell would be very useful in terms of encapsulation and being able to look at diabetes. We might write a program that looks like this, navigating this pathway. If I am a stem cell, I need to become endoderm. From endoderm, I need to become a progenitor cell. And then I need to go into the endocrine progenitors, and eventually I might be that beta cell. And I could write a series of conditional statements. If I'm given an external small FDA-approved molecule, express a sulfate regulator, and if I've, in that state, express the next regulator, express. And that's a very traditional way of thinking. I can coax a cell among these states. But I started by motivating that we want to build organoids, and organoids need to have all of the different cell types. So a program like this is inherently going to fail, as it's written to only produce one cell type. So we started thinking to ourselves, what can we learn from biology, and how can we actually navigate where we can coax cells among these different lineages in a kind of predictable, systematic way. And to do that, a few years ago, we really looked at kind of germline construction and what are the main sulfate regulators. So there's a lot of feedback control between nanogs, SOCs, the GATA factors, and HOXs. And a few talented postdocs in the lab who have since moved on came up with the following design. Let's just start with the simplest thing. Let's just see if we can make endoderm reliably, and let's see what happens from there. So we introduced a circuit alongside with Linda Griffith and Mo. If we have this DOX induction, let's change the stem cell into endoderm by expressing GATA6. But unlike many other features where you kind of add your cofactor in the media, let's deliver it in a heterogeneous way so that way the cells themselves have some bifurcation available to it. So we designed a lentivirus, as that was the state of our delivery method, and we introduced the circuitry where we can have our DOX-inducible GATA6, we engineer into our stem cell, and then we let the stem cell go. And what should happen is if we integrate this in multiple copies, each cell has a little different expression of GATA6, and indeed, we can see that where we have a heterogeneous expression of GATA6 based on our infection titer levels and the stem cells that we actually grew. And then we let that go. So transiently applying DOX for 5 days allowed us to actually start seeing differentiation. So at day 0, we have 2 populations of cells. We have our blue cells, which have our DOX engineered circuit, and we then have a red cell, which is just a iPSC engineered to be red. We induce with DOX for 5 days, and we see that our blue cells are still blue, but they turn green in relation to that GATA6 being expressed, and we still see that red. So in 2 dimensions, we are critically breaking symmetry of these 2 populations, allowing us to get heterogeneous expression of GATA6. We let that go a little bit longer, and one of the cool things is we actually saw first that high and medium GATA6 went to mesoendoderm, and then to endoderm and mesoderm was low, went to ectoderm, and we could characterize that by actually looking at your favorite immunomarkers. And what we found is we were actually generating, by having this bifurcation of GATA6 expression, all of these cells from our ectoderm, we were getting actually neurons and some kind of forebrain aspects. We were also seeing a large diversity of cell types that looked like a liver lineage. So with a hammer, but only for 5 days, we were able to actually layer a endogenous exogenous program. So DOX GATA6, a few days in, we're starting to see creation of really a structural functional thing from a single stem cell. It looked a little bit like a liver. In that way, we could see that there was some urea production, there was albumin production. So we got very excited. And we decided to, what can we do next with this system? So with some talented former postdocs in the lab, we decided to go into 3D. So right now we had been on 2D, and we see all of these precursor cells. And the way we did that is we take that same system, but we use all of the technologies that we've seen for spheroids and development of these spherical with the ultra-low adherence plates. And we actually then go from DOX from 0 to 6. We move DOX, we go to a synthetic defined animal-free media, APOL2, and we let these organoids develop. And what we saw is we still had that GATA6 expression, but something crazy happens when you go to 3 dimensions, is we actually started seeing that our hepatocytes from a day 40 organoid recapitulate that polygonal tessellation structure. And the thing that I think surprised us the most is we actually saw creation of structure where we have our hepatocytes here in yellow, but now we're getting actually the separation with our stellate cells, our endothelial cells, and we're recapitulating the fact that the liver is just a series of tunnels. And so at this point, we start calling ourselves plumbers, as we're just trying to figure out how do we get our arteries, our veins, and our bile ducts formed. And this, again, is surprising because all we've done so far is layer a simple circuit on top, and we now can see that we've generated a lot of cell types, but are these functional, and what really is happening? So we decided to go and dive in a little bit using all of our modern techniques and said, are we actually going through the differentiation pathway that we think is happening, and how could we actually study that? And are we actually differentiating correctly? So really cool, though, we can actually see that now these DA30 organoids have fully pervasive vasculature, and we can see the generation of these arteries. And so here's a day 6, here's a day 15, and then at day 30, we can see our hepatocytes, we can see our vasculature, we are seeing bile ducts, but are we actually making something that functions as a liver? So we dove back in and we did a little bit more characterization. So they're really big at day 70. They're some of the biggest organoids right now in the country. and we're seeing all of the cell types that we'd expect. So we see our endothelial cells, we see our hepatocytes, but we're also seeing cholangiocytes for our bile ducts, and we're making Kupffer cells. And so the biggest resident monocyte population are actually Kupffer cells that haven't been activated yet. And we are now really excited about kind of next generation immunotherapies with our liver. But we also went through and saw, are these actually functioning as a liver? And what we saw is if you compare our organoids to any one of our healthy adult livers, we're actually producing albumin and urea at the same concentrations and same turnover rates as a model system if we were to take a biopsy from any healthy liver patient. So now we were really starting to think about how do we apply this, because it looks like we have a liver at first approximation. So we went back through and we started thinking about, well, we've made urea, we've made albumin, we don't have the right structural specifications yet, but we have all of these other cell types, and we wanted to validate, are they actually developing correctly? Am I actually, just with this hammer, able to get a liver? Because if that's the case, that's remarkable. But if you've worked in biology, you know that it's never the case. So we employ single-cell RNA-seq. And so with a previous postdoc who's still at MIT in the Pekauer Institute now, we looked at our organoid development and single-cell generations and clustered all of our cells. And so now I just want to focus in on just our engineered iPSC all the way down to hepatocytes and really hone in on, are we making mature hepatocytes? So starting with our iPSC population in red at the very top of our single-cell RNA data, we can start characterizing where our genes turned on in this developmental pathway, and are they indicative of what we'd expect. So I have brachyury expression turning on in that beige population, and that's indicative of a mesoendoderm. I then have my SOX17, so I'm now making definitive endoderm. We have GATA6 control, which is all of our hepatic progenitors. And finally, we look at HNF4 alpha, which is the late stage transcription factor that is the kind of start of fetal hepatocyte development. So we've now identified these clusters, and we see that actually the map and the lineage tree that we believe we're doing is actually coming over. But there still seems to be a gap between what we'd expect when it comes to this last step of actually getting mature hepatocytes. So to do that, we went across the river to our friends in Merck, and we asked ourselves, how do we actually look at liver systems that they use in their pharma conventions? And one of the things that is the gold standard still is HepatoPac, a technology here from MIT, from Sangeeta Bhatia's group, as well as primary human hepatocytes. One of the things that was really interesting is very few people have characterized primary human hepatocytes across a wide set of patients that range in sex and age. Oftentimes, you're only looking at single or pooled donors. So with a pooled donor, you get an average measurement. Single donors, you always don't have the diversity. So we first set about characterizing HepatoPac and a large set of primary human hepatocytes to establish, for ADME function, any drug development, no one actually had a good sense of what is the variation of every CYP in the body per patient. So we generated some confidence intervals, and we looked then at 130 genes that were relevant for ADME function in our livers. And we're looking at then transcript number in primary human hepatocytes versus those in a day 35 organoid. And surprisingly enough, about 100 of these genes are expressing at levels comparable to a healthy adult liver in a day 35 organoid. And so just to really let that sink in, a 21 to 25-year-old process for really true maturation of a liver is happening in 35 days. And we were just very blown away by this. However, we're always going to try to make a better system, because that is what we do at MIT. And we noticed that we are not there yet. There's still enough genes that are not expressing correctly in these organoids. We wanted to why. In particular, if you've ever had to stop having grapefruit juice, one of the reasons is grapefruit actually inhibits the CYP3A4 pathway. And the cytochrome P450 3A4 variant is responsible for about 70 to 75% of all metabolism of most active pharma compounds we take. And unfortunately, our organoid, honestly, is pretty terrible. It's one of our lowest performing genes. So we started to ask ourselves, I'm a synthetic biologist, we are synthetic biologists, we should be able to fix this. And so that's what we had to do. I need to boost CYP3A4 function. I need to better understand how do I actually get my fetal hepatocyte into a mature hepatocyte. So to do that, we have to write a new program. So our original program is if there's docs, express GATA6, heterogeneity with a pulse, but now I need to introduce another circuit where I want to say if my cell is a hepatocyte, express different payloads, maturation factors, maybe just express CYP3A4 straight up to see that we can do this. And so over the last, one of the things though is we don't want to interrupt any of the other development. We have a good liver model. We have cholangiocytes, we have parasites, we have Kupffer cells. So we need to somehow improve the maturation without actually influencing any of the other cell types. So we need a very cell-specific, temporal-specific, as each one of these cells is happening in many cells. So we need to have a sensor that's actually particular just for that fetal hepatocyte. To do that, over the last few years, in the Weiss Lab, at MIT, and the SynBio Center, we've been building a lot of platform technologies on how do we sense cellular state. So I will introduce, this is from a few years ago, as well as a recent grad student who's now at Asimov, where we engineer microRNA sensors with the concept of RNA interference. If we take microRNA targets and append it to the 3' end of any coding region, we get the behavior where the microRNA binds, we knock down expression. And to get a high sensor, as we need to be able to sense low and high microRNAs, we introduce the concept of an inversion, where we take a repressor at the DNA level, add microRNA targets to it, and that repressor in turn binds and influences our payload. So in this case, when the microRNA is present, we get output. So we built then our microRNA sensors, and we've done a lot of work building cancer classifiers in the last decade, where we can make and or in any Boolean logic function for microRNA signatures. However, we tried this in stem cells for many years, and it just doesn't work. Because one of the things that's hard about stem cells is they're always going through epigenetic silencing and epigenetic destability. So if you look at actually these DNA circuits, they just don't work. So we needed to develop a new technology, and with the advent of all of this excitement around RNA, whether it's base editing, whether it's RNA vaccines, we'd also been working on a platform with two talented grad students, one who's already left and one who's finishing up soon, where we thought about can we use CRISPR and Cas enzymes in a non-traditional way. Instead of doing base editing, could we actually do an RNA regulation? And so in this case, we introduce our type 3 and type 4 systems, and we make something we call a persist off switch. So we have our DNA payloads that express at the RNA level, and then our endoribonuclease cuts its cognate hairpin, leading to destruction of that mRNA. Or we have a persist on motif, where now we actually have a triplet stabilizer on our RNA. We then have our endoribonuclease site, and we have RNA degradation domains. So now when that endoribonuclease cuts, it cuts off the degradation domains, allowing expression. So this is all happening at the post-translational level, and I won't get into it today, but there's been a lot of evidence that if we have things expressed continuously in iPSCs, it reduces silencing, as well as when you integrate into the genome, we use a lot of chromatin insulators to hopefully keep these from being silenced, as our differentiation pathways are going now for months. And if we actually think about like a deployable cell therapy, we might be on the order of years. So RNA silencing and DNA silencing, I think, is probably one of the hardest things and challenges left right now with stem cells. But now with these two technologies, we can combine them together with a friend of mine, Lei, who can't be here today. She's giving her own talk. We've been working the systems in stem cells where now we have our microRNA targets on the 3' end of our fluorescent proteins, and then we introduce our Cas or endoribonucleases, so it's at the RNA level. And what that looks like then is we can create circuits like this, where we actually have our endoribonuclease flanked by microRNA 122, which is specific for the liver. And in this case, if I am low, that is, I'm still in iPSC, I'm still in a differentiation pathway, I'm still not even a fetal hepatocyte yet, I am not expressing any of my payloads as Cas is on. Cas then, we have some fluorescent proteins for our help as it's still in development, but we are not getting our payloads, which is these transcription factors or CYP3A4. When I become a hepatocyte, I will now have my marker RNA 122 is high. I knock down Cas-C expression, and now we're getting those payloads. And so a generalizable, this is just a single endoribonuclease, a single microRNA, but we've shown combinatorial logic not in stem cells quite yet. So we go ahead and do this, but one of the things that's challenging is how do you actually optimize this? Because you have these relation curves between the amount of Cas-C and microRNA, but if we increase the payloads, and every patient's gonna have a slightly different microRNA signature, you get different behaviors, and it actually maps really, really complex to the point where you can't actually optimize this very easily. So we had to design optimization strategies of how do we actually get the behavior we want, because any one of these, if we change it, and it's a patient-specific manner, we need to get a new behavior. And so a few years ago, we invented a way where we can actually combinatorially scan space using the fact that when we transfect something, you have a lot of heterogeneity possible. And so in that, rather than doing a co-transfection, we introduced separate complexes, and I'm just showing you in 2 dimensions, where each cell now is gonna have a different integration amount and a different integration event in each one of these piggyback transposons. And then we generate these regions where you can now rank high, low, medium across 3 dimensions, and you end up with 27 cohorts we can probe with qPCR. One of the things, though, is we're really big on, we've shown structure, we've shown that we're getting cell types, but we really wanted to show function. So we turned to our friends at Merck again, and after showing that we were able to actually boost CYP3A4 function on this manifold space in 3 dimensions, does that actually relate to toxicology studies? And so we incubate our organoids with these substrates, we collect those samples, we use LC-MS, MS, and in turn we get these chromatograms where we can actually see, are we getting turnover indicative of what we'd expect? And indeed, when we look at our original GATA6 organoid Gen 0 to some of these new generation organoids with these increased payloads, I've short-circuited development, so CYP3A4 is there, and I've increased CYP3A4 expression and turnover of these substrates, all of which are FDA molecules currently in use. But then, because we have additional maturation factors, when we look at the battery of other CYP enzymes, we actually see increase across the board of metabolism of actual drug molecules. So we were super excited, as with fairly simple networks, we're able to actually build a better organoid by expressing things conditionally through the developmental processes. And from here, we're really excited, as I'm just mindful of the time, where we're starting to apply this across a large array of application spaces. So we've shown that we can make these liver organoids, that they're functional, we can symmetry break, we have conditional expression, and we can at least do some compound screening. But we're really excited about what the future brings, which is how do we actually get to transplantation? How do we encapsulate these, because they're patient-derived, they could be allogenic, they could be immunogenic, and how do we actually get to implantation stories where now we're actually growing a better liver? Which, why can't we grow a liver with better function, with different enzyme chemistry? And so as a little teaser, we've been working with NORA, the North American Research Alliance with BASF, where we've now taken our organoids and been looking at some compounds that failed their toxicology studies because the model systems they're using, animals and HepG2s, don't even have the enzymes needed to break down. And we've been starting to do analytics with them where we're able to take compounds that failed their screening and actually they're metabolized by our organoids, so we can see mode of actions that you just can't see in 2D or in mouse when it comes to human physiology. And the last thing that's really exciting is with Valerie over at BU Medical Center, we've been taking our organoids now and implanting them into mouse, into the kidney capsule. And what we're seeing is we actually can see human albumin being produced in these mice models, suggesting that our liver organoids are living and they're able to actually continue to perform. And more importantly, we actually can see them very tolerant. So up to 90 days now, we've been implanting our organoids and seeing that right now we're in the kidney capsule. We're still working on the surgical techniques to put them with a mouse liver or in a diseased mouse, where we actually see that our liver grows and grafts right on top of the kidney, starts to have vasculature join in between our existing vasculature and that of the mouse, and have actually albumin production and liver function. So with that, it's really exciting time, I think, to be in Kendall Square always, but particularly when you think about Linda Griffith, one of our collaborators now, 30 years ago, the first thing of just putting scaffold on a mouse and squirting some cells on it, and like structurally it looked like an ear, but functionally it's far from it. But now we're finally teasing out our first set of organoids that are actually functionally interesting, and really, I think, going to usher in, at first, a new kind of era of drug development, but I think we're really well on our way into organoid transplants and organ development. So with that, our group is wonderful, and I've been at MIT now for many, many years, and it's always been a pleasure to see all of our collaborators across industry, our funders, and a really great lab group right out of Tech Square. So with that, I will happily take questions.
Thank you, Deepak. That was an excellent presentation. So we got a couple of questions here. Can you see these from your end?
Okay, so let's start with what longitudinal monitoring intervention techniques. So one of the hardest things is getting a new drug development strategy approved is going to be a 10-year process. So we've been working with, one of the nice things about working with BASF is they are the largest chemical company in the world, and we have been slowly and steadily showing up now at the drug development congresses as regulation and all of the rules for actually a new clinical piece of the puzzle are identified. I think one of the hardest things up until maybe 10, 15 years ago that we've learned from synthetic biology is scientists need to be in the room with pharma and regulators. We need to be helping teach, educate, and usher in new technologies. And so with that, we've been working with now a consortium of humanizing drug development with MIT here, where we now have around 30 different pharma companies that come and listen to our techniques and give us strategies on, okay, we need to be able to interface with this robotic system. We need to actually be able to do 1 million organoids when you're only doing 1,000. So I think it's unclear like what's the best organoid going to be, but I was just at the M-Cells and Organoid Keystone last week, and we're starting to have NIST come into our conversations as well, is if we can just get a organoid-specific measure of urea, measure of albumin, measure of function, that's already how primary human hepatocytes and hepatopac are validated. So we just need to show that every organoid we make has that QC on it. So the organoids, the size, we do not have any Warburg effect or necrotic core, and there are still a couple different explanations we're trying to figure out. One of the reasons is because we have this full pervasive vascularization, and there is probably, it's not active perfusion yet. We are working with Roger Cam here at MIT on getting them actively perfused. But we don't have a good understanding of why there's no necrotic core, because most spheroids of single or double cell types, once you get above 500 microns, do die in the middle. And we're easily going to millimeters, and we're not seeing that. So it's unclear. We're trying to study it. One of the hard things has been just really understanding how much of our diffusive flow. So we're starting to work with chemical engineers to actually get better models of our pervasive. One of the cool things this summer is I have 4 students, and we're going to be actually using a very old technique using a chicken egg, which is one of the easiest way to perfuse an organoid and actually do a CAM assay where we put our organoids and really try to understand, because we're not seeing nutrient limitation, but we're not sure why. Yeah, for transplantation, and I will happily talk offline, we need to encapsulate, so we're starting encapsulation studies, or we need to make a cartridge, or we need to make a mesh, or we need to do something, because right now the FDA, there's a lot of studies with encapsulation in animal models, where you can actually put into the intraperitoneal cavity beta cells, and you don't have to worry about recovery. One of the biggest things I think on the road to transplantation is the FDA is going to require us to get our organoids back out. And the only way we can do that is to actually probably make a cartridge with an existing ability to connect to your vasculature, but have it encapsulated in some way. I think we might get to a point in maybe 20, 30 years where we've shown enough efficacy and safety data that we won't have to always recover our organoids, but to start, we are going to have to be able to recover them. I think so the benefits of organoids from scratch is that it's complementary. We don't know enough biology yet to definitively say that we're not going to 3D print scaffolds or decellularize some organoid and then add other cells on top. I think it's very much a bottom-up and top-down approach where it's going to be the marriage of actually 3D printing and using iPSCs, and for all we know, maybe introducing mRNA technologies so that way we can re-engineer our organoids at day 20, at day 30 with additional programming. I don't think it's going to be a monolithic approach, but I think for now we really just need to further the field of organoids and iPSC development, as that's going to be the only way we actually get like patient-specific organoids, because we'll never have to worry about rejection. And then, I don't know if you want to cut me off anytime. I can talk all hour. Oh yeah, okay, 10 seconds, awesome. Okay, so for transplantation, like clinical trial substitutes, I think are the easiest thing to really get behind, is we can do biology that other models cannot do. So I gave you 2 examples where now that we've increased CYP3A4 function, we are starting to like really go into a test suite of 100 different therapeutics that Merck Inc. has generated to see if we actually see the same turnover rates that they get from primary human hepatocyte. And then we're going to start thinking about, can we use unknown molecules or unknown variants? We've already started doing that with BASF in toxicology, where we have a couple different chemical compounds that they have not been able to figure out mode of action on that we're now putting onto our organoids, able to do mass spec and actually able to tease out new science of metabolomics of some of these compound classes. So I think that's just the easiest thing. In the future, we're somehow, I don't know if it's 30 years, I don't know if it's 60 years to true implants, but I think we are now just starting to scratch the surface this summer where nobody can really make good disease models of the liver. So if we have cirrhosis, we could imagine actually engineering our organoids to be a cirrhosis model from the start. And now we do drug development on them. By accident, we put LPS onto our organoids and actually saw inflammation. And by accident, that means like our organoids got contaminated, but then we did RNA-seq on it and actually saw increased inflammation. And so we're starting to now really think about how do we make disease models with this technology. And with ILP, they've been great as they've been connecting us to people to actually talk about what disease models should we even think about making.
Great, thank you very much, Deepak.